Good morning. For those of you here now, just to give you a bit of heads up, on Show My Homework, I have put on the assessment for the digestive system. It is going to be like a test where you fill it in, you return it to me, and by the end of the deadline, that's when I'll be marking all of it and then returning it to you along with feedback, just like we sort of do in class. So the purpose of this stream is to help you with that assessment. And now that assessment is only covering year nines, but like I said, it would be useful for anyone to take part in this. Okay, so just as a quick recap of what we did yesterday. Yesterday we mainly touched on all the different organs of the digestive system, and we also talked about what they did. Okay? So, to start off with, just as a quick recap, oops. We started with the mouth. That is where ingestion happens, right? You take things in, you eat it, and also mechanical digestion happens here because you're chewing it up, you're breaking it down into smaller pieces to make it easier later on for you to digest it chemically. Now, at the same time you're chewing it, you're mechanically digesting it, your salivary glands are producing saliva, and that saliva has two uses. The first use, if we remember, is to sort of coat the food, to wet it a little bit so it goes down a bit easier. The second thing is that saliva also contains that enzyme called amylase, and amylase breaks down starch into sugar. So a little bit of chemical digestion happens in your mouth. After that process, it goes down the esophagus, which is that muscular tube that squeezes food down that allows you to swallow upside down, and then from the esophagus it reaches your stomach. The stomach contains acid, but remember the acid doesn't actually break down the food, it just kills bacteria and makes it a good pH for anything that you want. It makes it a good pH for your protease enzyme, the one that digests protein, to be able to work the best it can. Because all enzymes, all the enzymes in your body have sort of like the best sort of pH, the best environment it works at. And protease, the one that digests protein, happens to like a really low pH acidic environment, so the stomach provides that so the protease can work as best as it can. But your small intestine, we don't want it to be super acidic when the food reaches the small intestine. And also the other enzymes involved don't really particularly like super low pH like protease does, so we want to neutralize that acid that's coming from the stomach a bit. So your liver makes this thing called bile, makes this substance called bile. It gets stored in the gallbladder and released into the small intestine. So you'll notice this little tube here that sends it over to the small intestine. Okay? So there's that tube that sends it to the small intestine. And that bile has two purposes. The first purpose is, remember, it breaks down large fatty droplets into smaller droplets of fat to make it easier for digestion. And it also neutralizes that acid that comes from the stomach. Now, in the small intestine, that is where a lot of more chemical digestion happens. So, for example, we've already covered the fact that protease digests protein in your stomach. Now, in the small intestine, we have all of the different food groups also being digested. So we have carbohydrates to digest carbohydrates. We have lipase to digest lipids, or aka fats. But the small intestine, remember, doesn't actually make its own enzymes. Those enzymes come from the pancreas, and the pancreas sends it to the small intestine. Small intestine also absorbs all of the food that has been digested. So all of the digested, broken down food, food that's already been broken down by the enzymes, that now can pass into the blood, that now can be dissolved in the blood, get absorbed in the small intestine through those structures, remember, known as the villi, just to make sure that we have a large surface area for as much movement of the food, much absorption as possible. After all of our nutrients get absorbed, then we move on to the large intestine where water gets reabsorbed, and then the solid that is left over gets stored in your rectum and then passes through the anus. Okay, so that's what we covered yesterday. Now we're going to just quickly recap the enzymes, because you are going to have to memorize the names of the enzymes. But remember, the name of the enzyme gives you a pretty good hint of what it digests. So lipids is our scientific word for fat. It is fat, it's the same as fat. It covers oils as well, but it's basically fats and oils. Lipids gets digested by lipase. Proteins get digested by protease. And carbohydrates gets digested by carbohydrase. And there's a special type of carbohydrates, carbohydrase, that we talked about earlier, that we can find in our mouth, called amylase, and that specifically digests starch. So those are the enzymes that you have to be aware of. Okay? Just because of time, I'm going to just briefly cover this next bit, because it goes into a lot of detail about the structures of the carbohydrates, lipids, and proteins. It'd be best if you sort of could look at Kerboodle for this, because it is sort of like a minor point of knowing the structure of all the different food groups. But it's not really the major key area that comes up again and again on GCSE. So this is one thing where it's like a cherry on top. So I'm going to skip this for now just because of time, and move on to specifically how enzymes work, how enzymes do its job. So enzymes are what we call catalysts. And you would have heard of this word before back in Key Stage 3. You would have heard of what catalysts are. Now, catalysts in general, you may have heard of catalysts in the case of like a catalytic converter in your car, things like that. So, a catalyst is anything that speeds up a chemical reaction. But it doesn't get used up. So basically, the chemical reaction happens. Okay, the chemical reaction happens, and it's almost like the catalyst is the facilitator, the one that sort of oversees the chemical reaction, but it's not part of it. If you think about it like a sort of a relationship, like there's a chemical reaction between one person and another person, they fall in love, it's all romantic. The catalyst is like the matchmaker, the wingman, the person who got them together in the first place. They sort of sped up the process of getting the two together, but they're not part of that relationship. That's sort of my analogy for what a catalyst is, right? It helps speed things up, but it's not actually really part of it. It doesn't actually involve itself in the chemical reaction. It just helps the reactants get together. And so, an enzyme is an example of a catalyst. So, in our case of digestion, our chemical reaction is the breaking down of food, it's the breaking down of starch into sugar, it's the breaking down of fats into its individual parts. So, those are all chemical reactions that are happening, and enzymes don't get involved in the actual reaction itself, but it helps speed things up. And now we're going to talk about how enzymes do that. So basically, we call enzymes biological catalysts because in chemistry, you're going to be talking about a lot of different types of catalysts that are like, just pure chemicals, like platinum even can be used as a catalyst sometimes. But enzymes, because it's in our body, we call them biological catalysts because they're specifically catalysts that help us, that help living things. And they have these areas called active sites where the chemical reaction happens. Now, this is a very sort of common diagram just to help you think about what happens. So here is the enzyme. This is my little sort of gray thing here. This is an enzyme. Now notice it has kind of like a really weird shape. That's because each enzyme has a shape that is specific to what thing it's trying to break down. The thing that we're trying to break down is known as the substrate. I know it's a lot of keywords right now, but let's pretend that the substrate is, we'll go with protein. Let's say this is a protein. That means this enzyme is protease. Now it has an active site, basically an area that is going to bind to the protein, that's going to connect with the protein, and notice that it's a perfect fit. This shape on the enzyme here perfectly fits this protein. And then when the protein fits, the enzyme breaks it apart and then releases the products. So at the end, the enzyme is still the same shape. It's not part of the actual chemical reaction. It was just a place to allow for that breaking apart to happen, to allow for that chemical reaction to happen. And this is known as lock and key theory, because if you imagine this enzyme instead had this bit filled in, well, its shape wouldn't fit our protein anymore. When you think of locks and keys, if you have like a lock, only a particular key can fit into it, right? Like if you have a house key, you're not going to take your house key, go to your neighbor's, and have your house key fit into your neighbor's keyhole. That's not how it works. The same goes for enzymes. Each individual enzyme has an active site, has an area that only fits a specific substance. That is why we have an enzyme for each different food group, right? Like protein gets digested by protease. Protease cannot go then and then try to digest a carbohydrate. That's not how it works. Each specific enzyme only fits a specific substance. And now just to give you an idea about the keywords, just to recap the keywords, the substrate is what is getting broken down. So you can think of it like the reactant in this chemical reaction. The active site is where it happens. And the enzyme substrate complex is just basically this bit where they've when they have connected. So when the substrate and the enzyme have connected together. Okay? Right. So once again, just because of time, have a look at Kerboodle for this, because this is just sort of let me give you a general overview rather than giving you the actual notes. So basically the enzymes in our body regulate metabolism, where when you eat food, you digest food, you use it for energy, and then you have to eat food again, and it's that cycle. So in your body, metabolism is basically just the sum of all the chemical reactions that need to be happening in your body for you to survive. Like for example, building, so growing, building muscle, anything like that, or even like building up proteins in your body, building up any substances in your body that you need. Changing, so all the changes that happen in your body, and breaking down the foods that you eat. So metabolism is just sort of like the sum of the chemical reactions that are happening in the body, and each of those chemical reactions, to make it happen faster, we need enzymes to do it. And we have loads and loads of different enzymes in our body that all do specific jobs because of lock and key theory. Because one enzyme there's no such thing as like a master enzyme like there are master keys. One enzyme does one specific job, another enzyme does another specific job. But enzymes can also sort of, you can think of it like die, but they're not like actually alive, but they can sort of break down. That's a better word for it. So they can break down. We call this breaking down denaturation. Basically, here is a picture of a normal enzyme, right? Lock and key. So the yellow bit here is our substrate. It's fitting into the green bit here. That's our enzyme. But let's say that we turned up the heat and we took our enzyme, we put it in a pot of water, and we started boiling it. Well, that enzyme's going to sort of break apart. Kind of like you know, anything breaks apart when you give it enough heat, and it becomes denatured, where if you notice, its active site now looks a little bit worse for wear. It looks a little bit messed up. And so it's kind of like if you melted the keyhole. Your key, your substrate, is no longer going to fit. So when it no longer fits, this enzyme no longer works. So the enzyme's shape slash active site no longer fits substrate, so can't do its job. That is why when you have a fever, why it's so worrying. If you have a fever and your temperature gets like a super high, um, where it's like in the dangerous territory. The reason why that super high fever is dangerous is because the enzymes in your body can't handle that heat, and they can become denatured. And when the enzymes in your body become denatured, then we're going to have a problem. So that is why you always try to, want to bring a fever down to make sure that your enzymes could still be working to keep your body functioning. Now, there's always going to be like the best scenario for an enzyme, and we call that optimum temperature. So like the best temperature for an enzyme. And that's when the enzyme works the fastest. Um, and then at higher temperatures it gets denatured. So if you have a look at this graph right now, on my x-axis is temperature. So from left to right we have increasing temperature, and then on my y-axis up here is the rate of reaction, basically how fast our chemical reactions are happening. If we want them to, happen as fast as possible, like for example in the case of digestion, we want to digest food as fast as possible. There's no point in drawing it out or anything like that. For enzymes to work the fastest, the best they can, there's usually going to be the best temperature. And the way you spot that is at the peak. So wherever you see the peak on this type of graph, where the rate of reaction is the highest, that temperature is going to be when the enzymes work the best. So in this case, it's about 40 degrees our enzymes work the best. But past that point, that's when the enzymes get denatured, and you'll see the speed of reaction fall rapidly. And the reason why is because now the enzymes stop working. They can no longer work, so that speed's going to rapidly drop. So when it comes to our enzymes, we want to make sure that our conditions in our body are best as possible for the enzymes to work. Now there are a few exceptions to the rule of what temperatures there are. Like for example, we have extremophiles. You may have heard about them before, but it's usually like bacteria or really small creatures that can live in really, really crazy conditions, like near volcano vents, things like that. Okay, the same way temperature affects enzymes, we can also have whether we can also have acids and alkalis affect enzymes. So usually enzymes have like an ideal pH or acidity that they work in. Remember, pH is just a measure of how acidic an environment is. And so usually there is going to be like the best pH for an enzyme. And just like temperature, if it gets too acidic or too alkali, that enzyme is just going to denature. So just like before, there's usually a graph, and that peak is what we call the optimum pH, so the best pH for our enzyme to work in. Okay? So just for you guys to be aware of what your responsibilities are, I have put on Show My Homework now, sort of like a quick test for you guys to do. Let me show you the test. It is on Show My Homework, and there should be a section on Show My Homework for you to attach your test as a file. If you guys could do me a favor just to make it easier for me to mark, like you could print this out, write on it, and then take a picture of it, but I recommend opening it as a Word document and filling it in as a Word document, like typing your answers and then just attaching it again. So just to give you an idea, you've already seen this, so I'm not like spoiling anything. This is already on Show My Homework. Question 1, the diagram shows part of the human digestive system. Naming the parts. So we just talked about all of the different parts at the start of this video. We talked about it yesterday. For you to name all of the different parts, 4 marks for it. Be aware of like sort of what the arrows are pointing to. I know it could be a little bit hard to tell, but try the best you can. A student has eaten a steak for dinner. The steak contains protein and fat. Describe how protein is digested. So what I'm looking for there is, for example, because we're talking about protein specifically, what enzyme digests protein? And it's 3 marks. So try to get into a bit more detail, starting from ingestion. And then explain 2 ways in which the body helps to in which bile helps to digest fat. So we talked about one way in huge detail, and then there's another way that we sort of touched upon, but I want you guys to sort of to use logic to talk about what the bile does and how it helps. And then we have some experiment analysis. The due date, because I put it on yesterday, it's a 2 week date. So the due date is on the 4th of May. So you have plenty of time, but obviously I recommend, just getting it done and out the way as quick as possible so you don't have to worry about it. The next question is experiment analysis. Name the food substance that amylase breaks down. Be a bit specific about it. Suggest what happens to the breakdown of the substance when food reaches reaches the stomach. Use information from the table to help you answer this question. So basically, I want you to look at the table and we have amylase activity in arbitrary units. Arbitrary units means like I don't care about the units of amylase activity, just sort of tell me what's going on. So as pH in the stomach, goes down, what do you notice? What do you notice happening in the table? Name a type of enzyme that digests stains containing fats, and then some other graph work. So analyzing the graph as best as you can. Describe the effect of increasing the temperature on the time taken by the detergent to remove the stain. So look at the graph and describe it as best as you can. Is the new detergent works at a lower temperature? Is it likely to be more environmentally friendly? So this is like a logic-based problem rather than a memorization problem. Neither detergent works well at 60 degrees. Explain why. So what we just talked about, have a think about enzymes, denaturation, and all of that. Okay? So you should have all the materials to do the test already. If you don't remember, you can look back on the previous videos because they've all been recorded. And just do the best you can. Remember, always look at the amount of marks something's worth to try to make sure that you've said enough for the question. So for example, if something's worth 2 marks, you have to say 2 things, okay? And then on Show My Homework, you can attach it as a file, but if you're stuck, if you can't find that option, feel free to email me directly as an attachment. That's fine. So this is due on the 4th of May. Okay, right. So I recommend you get that done now, just so then it's out of the way, and then we'll be done with the digestive system. All right. Any other questions about this? Or are we all okay and clear on what we're doing? Right, awesome. So remember, if you're stuck on attaching it as a file on Show My Homework, email it to me directly. My email is actually right up there, the top corner, yac@ydeen.gloss.sch.uk. So make sure you get that to me by the 4th of May, but the sooner you do it, the better, because if you leave it for too long, you might forget. Okay, have a good rest of the day, everyone. Hope you will all enjoy the Easter break and hope you're all taking care of yourselves. I'll see you